Welcome back friends in one more video on 7th online classes. And this is the second video lecture on experimental biology. In this video, we will learn how to measure a cell size by micrometry. But before we start our lecture, we must know that measurement of the dimension of cell is done under microscope with the help of two microscale which are known as micrometers. Both the micrometers have microscopic marking on their surface. One of them is known as stage micrometer. It is a special glass slide which is clipped to the stage of the microscope. It has standard marking on its surface which are generally 10 micrometer apart. Or we can say that each mark on the stage micrometer is 10 micrometer away from each other. The other micrometer is known as ocular micrometer. It is a circular glass disc which fits into the circular shelf inside the eyepiece of a microscope. It has arbitrary marking on its surface. However, the distance between the two adjacent marks is constant for a particular ocular micrometer. We should remember this calibration should be done separately, not only for the different power of objective lens as well as for the same power for different microscope, because it may vary with the microscope as well. How to use a stage micrometer? As you can see, in the center of this glass slide there is a circular structure, and within that circular structure there are different markings for stage micrometer. To observe those markings, we have to fix it in a microscope. Just like a slide, we can fix it and observe it, and when we will observe it, we can see that those markings now are clearly visible under a light microscope. And in this experiment, the marking is from 0 to 100. How to fix a ocular micrometer? This is a circular glass-like structure. As we have already discussed that it also have arbitrary marking on its surface and which cannot be seen by our naked eyes. For that we have to fix this circular glass in the eyepiece. Fixing of ocular micrometer also may vary from microscope to microscope. As you can see, in this microscope, eyepiece has been removed from the microscope, and with the help of a cap, we can keep our circular ocular micrometer inside the eyepiece, and then this eyepiece is replaced in the microscope. If we will observe it, we can see that there are also marking which is clearly visible now. We can also rotate this scale by rotating the eyepiece so that we can orient it in our required orientation. How to calibrate the measurement for objective lens? For that, the required objective is taken to position. The required objective is that using which the whole cell can be viewed, and it covers the microscopic field to the maximum possible extent. With the required objective in position, the mechanical stage is moved so that a line on the stage micrometer coincide with the line of ocular micrometer. Then another line is searched on the ocular micrometer which coincides with another line on the stage micrometer. The number of division between the coinciding lines is counted for both the micrometer. And with the help of this table, the calibration factor for the objective micrometer can calculate it. We have to calibrate the different calibration factor for different objective lens as well as for different microscope. These are the photographs where you can see or you can try to calculate the calibration factor for 10x as well as 40x of a specific microscope. Now we will learn how to measure a cell slide with this calibration factor. For this we have to replace state micrometer with the slides. We have to use the same objective lens for which we have already calculated the calibration factor. Now you can see the marking of objective lens can be seen along with the material. By rotating the eyepiece lens, we can arrange or we can orient the micrometer in our required direction. As you can see, we have rotated our eyepiece so that measurement can be taken for width of the cells as well as for the length of a cell. Now, the number of ocular division covered by the cell is counted by viewing through the eyepiece. It can be used not only for the length but also for the width of a cell or a diameter of the cell. Depending upon our requirement, we can use different power of objective lens to measure the cell size. As you can see in these two photographs, 10x power has been used to measure the cell size when we are trying to measure the length of a cell, whereas 40x is recommended when we are trying to measure the width of the same cell. Not only a plant cell but also an animal cell can also be used. As you can see, this photograph is of human chick cell and 40x has been used. With the same calibration factor, we can also measure the diameter of these cells. Here one thing we should remember, that whether it's a plant cell or an animal cell or any other microorganism, we should not measure a single cell only. For a better result, we should use at least 3 different cells, and their cells should be randomly selected. Then only we can give a better idea about the cell size. Finally, when we are ready with the number of ocular division covered by the 3 different cells, we can use this measurement table to calculate the exact cell size as well as to calculate the average cell size. Cell size can be calculated by multiplying the number of ocular division covered by the cell and multiplying it by the calibration factor, which we have already calculated in a calibration table. So my dear friend, this was all about how to measure a cell size by using stage and ocular micrometer. In the next video, we will discuss the effect of temperature and organic solvent in the membrane permeability. Till then, like and share my video, subscribe my channel and press the bell icon so that you will get notified whenever new video is posted on my YouTube channel. Thank you. Jai Hind. Bye bye.